All right, let's get started. Good morning, good afternoon, good evening, everybody. Thank you for joining us today. We're excited to share a good story with you between DNAnexus, Regeneron, the UK Biobank, and NVIDIA around accelerating our time to discovery with whole exome sequencing on the UKB's research analysis platform. So I just want to introduce myself. My name is Phil Sweeney. I look after our alliance partners here at DNAnexus, and I'm going to introduce Harry Clifford to you. He is our moderator for today's session. Harry?
Hi, thank you, Phil. Yep, super excited to be here. I work for NVIDIA. I'm a product architect. So I sort of sit somewhere between the external partners and working with the internal engineering teams. As a bioinformatician by training, super excited to see this webinar come together. We've got some really cool topics today, from big data through to accelerated analysis, with some really great speakers. So yes, I'm moderating, including the Q&A at the end, so please do submit questions in the Q&A tab throughout the session, and we'll do our best to get to those at the end. And yeah, if you could just start the questions with the name of who you are directing it to, that would be very helpful for me, please and thanks. So first up today, we'll be hearing from Mark Effingham, who will be giving us an overview of UK Biobank and its mission. Mark's the CEO of UK Biobank, has an extensive experience in technical strategy and IT delivery in health informatics and big data. So fantastic, it'll be great to hear from him, very excited for that. After Mark, we're going to be hearing from Will Salerno from Regeneron on the analysis of the 450,000 UK Biobank participants. Will is Senior Director of Genome Sequencing Informatics at Regeneron, and they work on improving patient care using genomic approaches for drug discovery and development. So his group performs all of the primary and secondary analysis of the genomic data. And at over 500,000 samples a year, that takes some serious effort and of scaling and pace. So looking forward to that as well. Next up will be NVIDIA's own Tim Harkins, who will be talking about the accelerated framework Parabricks, which was used as part of this fantastic achievement. So Tim is product manager for Parabricks, has an incredible background in bringing disruptive genomic technologies to the market, ranging all the way from real-time PCR to microarrays to next-gen sequencing, and comes with a wealth of experience, over 20 years in industry and nearly 70 peer-reviewed publications. So always fantastic to hear Tim speak. And last but by no means least, we'd hear from Ben Busby of DNAnexus with a walkthrough for us on how to rerun the analytical pipeline. So Ben works on prototyping scientific data integration and complex compute environments, including for the users of UK Biobank. He and his group have experience in software development across omics, imaging, machine learning, and several other fields. So it'll be great to hear from him as well. And with that, I will pass things over to Mark, giving us the overview of UK Biobank and its mission. Take it away, Mark.
Excellent. Thank you, Harry. So Mark Effingham, I'm Deputy CEO for UK Biobank. I'm delighted to be here today with colleagues from Regeneron, NVIDIA and DNAnexus, using this first section just to share a little bit about the kind of background to UK Biobank to those who may not be so familiar. And before Will, Tim and Ben get into the really material and exciting parts of the session today. So as a bit of background, UK Biobank is a prospective cohort study that recruited 500,000 participants between 2006 and 2010. Each of those participants came to one of a number of assessment centres that were set up across the UK to get a good mix of diversity across age, gender and ethnicity. It's entirely altruistic research, as participants didn't get any feedback. They really are contributing information about themselves that will create a resource that allows researchers to understand the genetic and environmental determinants of disease, and really to help improve the health of future generations. So each participant came to an assessment centre. They answered a lot of questions about their health and social background. They provided a number of physical measures such as height, weight, grip strength, and they provided a number of biological samples, the majority of which went into long-term storage for future assays. Now, importantly, they provided consent such that we could follow their health outcomes by linking to a vast range of healthcare data across the UK provided by predominantly the NHS, but also to allow us to recontact participants to take part in additional enhancements, such as our project to image up to 100,000 participants, and to undertake additional things like web-based questionnaires, which we run on a regular basis. Now, UK Biobank has always been known for its breadth of data that it's collected and curated, its depth, given the large number of participants that allow substantial numbers to be created for case control studies to provide the right statistical power to allow good quality research to be undertaken. But it's also now been known for its duration, given we have more than 10 years of follow-up data available on each participant. So who is using this resource? Well, there's now over 25,000 approved researchers using the resource today, and it is growing exponentially. In the last year alone, there's been more than 8,000 new researchers, and we don't see that growth declining so far. There's over 3,000, well, over 2,500 applications. We're almost approaching the 3,000th approved access application. But if you look over the last few years, the demographics about where researchers are coming from has really changed over time. So 5 years ago, roughly half of all researchers were coming from the UK and the remainder from outside of the UK. If we look at where we are at the end of 2021, it's now 4/5 of all new researchers are coming from outside of the UK. I think that was largely fueled by the release of the genotyping and imputation data in 2017, which really increased visibility of the resource on an international scale. So why are researchers coming to use the UK Biobank resource? And it is back to that breadth of data, and particularly the genetic characterization of the participants in the resource available today. And as mentioned, that started with the release of the genotyping and imputation data back in 2017. We extracted a DNA sample from one of the buffy coat samples provided by each participant and used an array-based chip from Informatics to measure 850,000 markers from across the genome that were known to be associated particularly with diseases that affect us in later life. We worked closely with the Wellcome Trust Centre for Human Genetics to create a high-quality imputation dataset that takes those 850,000 markers and expands it to about 90 million markers for researchers to start to look at as part of their analyses. And already these data have been used on a very wide scale. The vast majority of all access applications to Biobank request genetic data as part of their analyses. And you can see today there's a large range of outputs already available. For example, the outputs of various association studies and summary statistics, which you can find in various public repositories. Now, something of note, I'm sure will be of interest to this community, will be that we're shortly making available two further new imputation data sets. So researchers, for example, in Michigan, have used the TopMed panel to create a new imputation data set. And additionally, researchers here in the UK have used a reference panel that's been generated from the 100,000 Genomes project to again create a further imputation data set, roughly up to about 300 million markers. And again, we'll be making these available in the very near future. I'm sure they'll be well received by the research community. But in addition to all of the research outputs that we're seeing through scientific papers and things like association to the outputs, I mean, this really has been a transformative resource, and that has opened up entirely new areas of science to be developed. And a really good example here is the whole domain of the derivation of polygenic risk scores, which really has emerged off the back of these very large-scale data sets being made available within the UK Biobank study. And what's really pleasing to see is how these polygenic risk scores, which assess an individual's propensity to develop a particular disease, such as diabetes or cardiovascular disease, based on small changes across a number of genes. These PRS risk models are now finding a way into frontline clinical care, and only recently have been incorporated into an over 40s check here in the UK. But the focus for this webinar today is on the sequence data, and particularly the whole exome sequencing data, which we'll talk about in more detail shortly. Biobank's been very fortunate that we've had industry consortia coming together to invest in these large-scale sample assays. And here for this project, it's a consortium led by Regeneron that has undertaken sequencing of the exome, the 2% of the genome that codes for proteins, and that is believed to be the most informative, particularly for drug discovery research. Now, some landmark releases last year where we made available 450,000 exomes in around about October, and all of that data is available on the platform today for researchers to use as part of their projects. And again, we'll talk about that in more detail shortly. A further consortium has formed with additional support from UK government and from charities to undertake whole genome sequencing on all 500,000 participants. This project, again, has proceeded really well. The sequencing has now completed. There's a lot of work underway to do QC and undertaking joint calling of a data set of this scale. And these data on all 500,000 participants will be available in the early part of next year. But already 200,000 genomes are available to approved researchers on the research analysis platform today. But as well as all of that genetic data that is used to characterize participants, it really sits with all of the other data in the biobank resource, both that collected as part of the baseline assessment, but additional data sets that have been released over time. And just a few examples from data that have been released in the last 12 months. So web-based questionnaire data, we tend to do a couple of web-based questionnaires over the course of a year to elicit information from participants about domains that we can't get, for example, from the likes of linked healthcare records. So a really good example last year was making available data on about 170,000 participants relating to pain. Additional sample assays. So two here to talk about telomere lengths, which was a project led by a team of researchers at the University of Leicester to undertake telomere measurements on all 500,000 participants. And all of that data was made available in March last year. And a further sample access project, this time led by Nightingale Health, who've used their NMR-based assay to measure around 250 metabolomic markers. This has been undertaken on the whole cohort, and we made available the first set of measures for around 120,000 participants in March last year, and with more to come. Now, I mentioned Biobank is running a program to invite back up to 100,000 participants to undergo comprehensive imaging. And it really put UK Biobank in a very unique position that it had imaging data on around 45,000 participants prior to the pandemic. This has allowed us over the course of the last 12 months to invite back around 2,000 participants, 1,000 who we were able to identify as having had COVID, and 1,000 matched controls who hadn't, to invite them back to our imaging assessment centres to be re-imaged and to create a unique data set that will allow researchers to examine the impact of COVID on particularly internal body organs. And already today, there are data sets for 2,000 participants available, and we're seeing really interesting papers already emerging, such as a paper by a team of researchers at University of Oxford, who've identified changes in brain tissue as a result of the COVID infection. And I'm sure we'll see many more interesting papers to come. As well as the additional data releases from the likes of assay measures, from imaging, web-based questionnaires. As I mentioned, we do collect linked health record data. And over the last 2 years, particularly in support of research efforts into COVID, we've been trying to increase the frequency of those refreshes. And we've been bringing in regular updates of the likes of primary care data, of PCR testing and vaccination data, as well as other linked healthcare record data over that time. One of our focus areas for the next few years ahead is to look at how we expand the range of additional linkages that we can incorporate into the resource going forward. And so my last slide before handing over to Will is just to give a bit of a sense of, in addition to all the data that's already there today, what else is coming down the line this year. Now, we have, in support of the COVID pandemic, we ran a seroprevalence study where we invited 20,000 participants, including children and grandchildren, to take part in a 6-month project where we invited them to provide a monthly blood measure, and we undertook an antibody-based assay to look at seroprevalence over that period of time. We are just finishing a project to collect a 7th sample some 12 months later, and all of these data, together with questionnaire data, will be available in the next few months. In addition, we ran a project where we invited the whole cohort to take part in a seroprevalence study where we distributed antibody-based lateral flow tests to look at seroprevalence across the whole cohort. And around 200,000 participants took part in that. And actually, these data are now available in the resource as part of the most recent release just last weekend. I mentioned about web-based questionnaires. We do typically 2 a year. We already have one underway right now, which is looking at a second cognitive function questionnaire, and that's currently being completed. We have additional questionnaires in plan for the remainder of this year, looking at long COVID, other neuro issues, and sleep. And again, once those questionnaires have been completed, those data will become available in due course. And then lastly, just to talk about proteomics, given all that genetic characterization, the focus now is on how we move beyond the genomics and looking at other omic areas. I mentioned already the Nightingale Health Project looking at metabolomic measures, but there is a new consortium that's being formed to undertake a set of proteomic measures using the Olink panel, and that project is underway. So using that Olink assay, measures for about 3,000 circulating biomarkers will shortly become available in the resource for an initial 50,000 participants. We expect those data to be available in the latter part of this year, and we're looking forward to that being extended to all 500,000 participants over the time ahead. So lots of exciting things to come to, but I'll hand back to Harry to the next part of the webinar. Thank you.
Thank you so much, Mark. That is such an exciting and unique data resource. I'm sure many scientists, myself included, can't wait to dive into that. So, and equally exciting data still to come by the looks of things. So absolutely fantastic. Next up is Will Salerno of Regeneron, so covering the analysis of the 450,000 UK Biobank participants. Over to you, Will.
Great. Thanks, Harry. Thanks, everyone, for joining today. And thanks, Mark, for that introduction. What I'm going to get into today is what we did at the Regeneron Genetics Center on behalf of the UK Biobank Exome Sequencing Consortium, which Mark referenced as a group of industry partners who came together to work with UK Biobank to make this data set available. And at the RGC, we did the work to do the sequencing, and my group did the processing and provisioning of these data. So I'm very glad to have the opportunity today to talk with all of you about some of the specifics of how we went through processing these data, the types of applications that we used, and how those are being made available on the RAP, which Ben will get into later. But I wanted to start with this idea that our goal at the RGC and with the Exome Sequencing Consortium is really to have this capital, fair, and equitable informatics, to make sure that the data that we generate are findable, accessible, interoperable, reusable, that the informatics that we do are reproducible, transparent. The idea being that this is not just a data set that people will consume just the outputs, but maybe want to reanalyze or harmonize with their own data. And we want to make that as easy and transparent as possible. So working with UK Biobank, with the UKB RAP team, with NVIDIA, to make sure that the methods that we used in our production to generate these data are identical to what's available on the RAP, and that there's no lag between how we generated the data and what's available to the community, both in terms of just understanding the code, but also the ability to execute at scale. So that's what's really the driving motivation behind all this work that we did to make these tools available to the community. And hopefully the slides today will gives them insight into exactly what we did. So the focus of these slides is just on how we process these exome data. And I'm going to walk through not every detail here, but be glad to answer questions at the end. People who are familiar with next-gen sequencing, it's pretty straightforward. We start with the FASTQs, which are the raw reads. And for the 450K release, it was 454,000 and change samples. We map those with the OQFE protocol, and I'll go through each of these steps in more detail. And that generates the CRAM file. So these are all the raw reads, lossless from the FASTQ, mapped to the GRCH38 reference in an all-to-where manner, retaining all the original quality scores. Gives you about a 24 gig file per sample. Once you have mapped to that reference, we've called variants, small variants, SNVs and small indels with deep variant using the Parabricks platform, which I'll get into as well. And a GVCF, for those of you who aren't familiar, it's a row-based file where every row is a variant allele, but there are also these binned reference blocks. So it captures information not just about the variants that are observed in an individual or a sample, but also for the stretches that don't contain variation, what kind of coverage do you see there? So we can say, do we know this is reference homozygous at a given position, or maybe there wasn't coverage at that position? And those files are about 800 megs a sample. And we have one of those GVCFs for each sample within the cohort that we were looking at. And those are combined through a joint genotyping method, GLNexus, into an aggregate genotype matrix. And that's our data release. And we represent that as a PVCF or a multi-sample VCF. We also have PLINK and BGen formats. And there's about 11 million variants within the exome target capture region. So like Mark indicated, we're looking at the protein coding regions. It's about 38 megs of genomic sequence, 11 million variants in those regions. We do also call variants in the flanking regions. They are of interest for different types of studies. So the total number of variants in this data release is about 26.5 million, but 11 million of those are in sort of the high confidence target regions that we were going after. So what I'll do now is I'll walk through each of these steps, explain some of our motivations and the tools behind them. The first step is this OQFE mapping. And what I've put here are links, QR codes that you can scan or screenshot and find these papers or resources, because there's a lot of really useful detail in all of these links that you might want to be interested in. But just to hit the high points, the OQFE mapping protocol, it's deterministic and all-to-where mapping to GRCH38. If you run the protocol twice on the same input, you'll get the same output, which is really useful. A lot of what we've done for this mapping step is with the idea that we want to make the data as insensitive to noise as possible. We want to make it as easy to harmonize with other data, and we want to make sure that the method is reproducible and consistent with how other large-scale data sets, for example, how the UK Biobank whole genomes are being processed. So that's why we include things like primary and supplementary duplicate marking, which is important in whole genome sequencing for structural variant detection. But we also recognize that these files that we generate, these CRAMs, they're going to persist for a long time. Mark mentioned the duration of this study, that at some point there might be a different reference people might want to use in the long run, and they want to reanalyze the raw reads. So one of our primary considerations when developing this OQFE protocol was that the CRAMs are lossless. They retain the original quality scores. You can recover the original FASTQ from these CRAMs, including all the unmapped reads. There's no data that we sequence that's not in these CRAMs. And I think that's important. It's thinking about these CRAMs from a long-term persistent resource of important data. In order to generate these CRAMs in this manner, we have this protocol, the OQFE protocol, which is described in the links on the right. But we've also worked with the UKB RAP team internally and externally to develop both a Docker, which is publicly available from DNAnexus, and a application that sits on the RAP that is a push button from FASTQ to an OQFE CRAM. And we've designed this so everything you need to generate an OQFE CRAM is embedded in this Docker. You just give it a FASTQ, press the button, and it'll spit out an OQFE CRAM. Like I said, it's preloaded, and you can also start from an arbitrary CRAM or BAM as an input. So if you already have sequencing data that has been mapped to a reference, doesn't matter what reference it is, you can just provide that as an input, the app will roll it back to a fast queue and give you an OQFE version of the CRAM. So it really is designed to make as easy as possible for you to take any existing data you have and generate a CRAM that's going to be as similar as possible to what was done for the UK Biobank exomes. Once we have those CRAMs, we need to generate our variant calls. And what we did at the RGC in order to get the best calls was that we worked with the deep variant team. Deep variant is a deep learning based small variant caller, and the links below give you a lot of background. There's a ton of interesting work out there if you're interested. We took the UK Biobank data and we retrained our own model for these data specific, just to eke out that extra bit of quality, which you can see on the right, how our custom model compared to the out-of-the-box deep variant model. This was done using version 0.10. All that you require as an input is the CRAM, the model, the target bed file, which just tells you what regions you're going to call the variants in, and the reference genome, which helps with the normalization of indel variants. And your output is a single GVCF, and you get these GQ binned reference blocks. Just to be clear, we did do this custom model for our processing. That model is available both on the wrap and on the publication for the OQFE protocol. So that is something that is available. If people are interested in doing their own retraining on their own data, all of these are described in various documentations that are linked here. There's lots of strategies, but we are also confident that using our model or the out-of-the-box model is going to be applicable to the vast majority of data that people would bring into the platform. So if you're more interested in doing your own variant calling, please do look into these links. There's lots of great resources. Once we've generated those GVCFs, well, actually, let me step back. Deep Variant is open source software. And one of the great things that we did with the Parabricks team at NVIDIA is to accelerate that open source software in a way that was really as close as I feel as possible to get to the open source version. Because we're operating at such a large scale, time is a consideration, the ability to iterate and refine protocols is a consideration. We really think about the ability to do our research at scale. So what you can see on the right are just some data comparing both the runtime and the cost. And all the costs I present today are using, Google AWS list price in US East 1 and grab the spot instances. So these are just a proxy for whatever your pricing is going to be. And we know that on the UKB wrap, there's very competitive pricing that the UK Biobank has enabled working with AWS. So your costs may look a bit different, but the relative comparisons will hold. That the Parabricks acceleration really does save time and it saves money with the parameters that were run, in the top left here. But what I do want to emphasize is that because we're operating at scale for this project and we want to generate these data quickly and get them into the hands of researchers, we leverage accelerated methods. We don't want there to be a gap between our accelerated methods and what's available to people open source. And we've tackled that two ways. One is working with the NVIDIA and Parabricks team to make sure that the platform is available to UKB wrap users. And Tim and Ben are going to go into that more. But we also did a lot of bioinformatics work with the Parabricks team and their deep variant implementation to ensure that the accelerated version is as close as possible to the open source version. And so just as one example of how we evaluated this, we took 100 UK Biobank samples. We ran it both ways with the open source deep variant and with the Parabricks accelerated. We got 7.5 million sample level variants called with 100% concordance of alleles that were identified. Because of hardware differences, there are slight rounding differences between the genotype quality scores. That resulted in one zygosity mismatch out of 7.5 million variants. But overall, 81% of our calls had the exact same genotype quality. And for the 19% that had different genotypes, they differed by less than one, actually less than half on a Phred scale. So this really is as close as we can get between two different platforms. And we're really grateful to all the teams that work to make sure that the open source version and the accelerated version are as similar as possible. But people on the wrap don't need to worry about that because this Parabricks platform has been made available. Once we have the ability to run at scale. I just wanted to give an example. Again, these are some links to some of the things that we've talked about. But when we first implemented the OQFE pipeline, it was our second iteration of a pipeline. We got really great feedback from the UK Biobank users about how we could better update the pipeline. So we did that. And one thing we had to do was recall the first 50,000 samples once we had done the remapping. And this is just an example of what that looked like. To take about 50,000 samples, 50,000 crams, which is 40 terabytes of data, and recall with Parabricks deep variant on all of them. It was under 2 days for under $5,000, again, using those AWS spot prices. So the point is that doing even this large scale reprocessing and variant calling can be done quickly and relatively inexpensively, which really does, again, allow you to do your analysis and reanalysis at the scale of the data, not worry about subsetting. Once we have all of these GVCFs, we do a joint genotyping step with DNAnexus. What DNAnexus does is take all of the GVCFs and it harmonizes the alleles. That way, if indels look a little different or SNPs look a little different based on how they were called, they get harmonized across all samples so that you have a squared off matrix. It also leverages those reference blocks that we referenced before, the GQ-bind reference blocks, so that for every sample and every allele observed, you have some observation, whether there's a variant allele, whether there's a reference homozygous, or whether there were missing data. DNAnexus is open source and it's available on the UKB wrap. It is parallelizable by genomic region. So when we think about scale testing it, we're really thinking about the numbers of samples we can do, because you can always parallelize by breaking the genome into chunks. But DNAnexus looks at all the samples simultaneously, and that's what we really have to test with the scaling. I think when I put this slide together, our team, led by Sheldon Bai, had tested this up to 800,000. Since then, we've been tested it up to 1.4 million. So we're very confident that this will scale to a large number of samples. And even though we've tested it on exomes, we don't anticipate any issues with the genomes, because again, it's just a parallelization question. And again, as an example, for the UK Biobank, when we did the 200K release and we pulled all of this together with DNAnexus, it took us about 2.5 days using 4 batches, again, less than $5,000. If you wanted to parallelize more, you could cut that time down. The cost will remain stable. It was about 50,000 core hours, though. But again, this is relatively large-scale genomics that we're doing very quickly and being able to turn these things around. The outputs are PVCFs, which can be turned into plinks, BGens, and PGens. There's a lot of documentation on the wrap that will walk people through that. So that's the high level, with a little bit of detail, I guess, on the different steps. And the reason I went through these mostly was that I want people to understand how we did this, how the efforts we put in to make them transparent can really help the user community access these data. But also, it's not just a matter of being able to reproduce what we did or understand what we did for this production release, but we want to enable users to combine your own data with these UK Biobank exomes, either if it's just data harmonization, make sure that you're looking at the same reference and the same variants, looking at case control cohorts, if you have cases and you want to use UK Biobank as controls and pull those together into a joint set, or if you want to assess other types of variation or other variants in UK Biobank. Maybe there are variants you observe in your data and you want to understand whether they're observed in the UK Biobank data by recalling. You can do all of that with the tools that are provided and on the UK Biobank wrap. 3 easy steps. Start with your data, upload it to the research analysis platform. And I put an asterisk there because you could put up your BAMs or CRAMs or FASTQs and run it through the whole pipeline. If you already have OQFE CRAMs or you have GVCFs, you can start there. You upload the data you're starting with, you run the balance of that pipeline on the UK Biobank wrap, which Ben is going to talk about. And then you run DNAnexus to merge everything. And when you run that DNAnexus step, you do have the opportunity to provide a bed file or a set of regions. So maybe you're not interested in looking at the entire exome. You're really just interested in a handful of genes or the HLA region or whatever it might be. You can restrict that combination step with DNAnexus to the regions that you care about. That might save time, that might save cost. And what you'll get out is that harmonized project level or multi-sample VCF, which can be converted into plinks or BGens. You can annotate it, use that as a substrate for your downstream analysis. So I really appreciate everyone taking the time to join today. I hope that this detail enables people to not just understand, again, how we generated the data, but also how you can use these tools to merge the UK Biobank data with your own and really use the UK Biobank exomes as a multiplier for your own science. So I appreciate everyone's time and hand it back to Harry so we can get on with this.
Thank you so much, Will. As I said before, this type of work takes serious efforts in scaling, so it's incredibly impressive stuff. Thank you. Thanks to the participants who are putting in questions as well. Keep on going. Looking forward to asking these at the end. As an FYI, because we've been asked this a couple of times, the recording and the slides from the session are going to be circulated by DNAnexus afterwards, so no worries there. It'll also be posted to the Resource Center on the DNAnexus webpage. And with that, I will hand it over to Tim Harkins, who will be talking about the accelerated framework from NVIDIA, which is Clara Parabricks. Over to you, Tim.
Excellent. Thanks, Harry, and thanks everyone for joining in today. Always exciting to be associated with such a landmark study. So just as the project we're talking about today, the amount of data volumes that are coming out are just fantastic. 10, 20 years ago, we were dealing with gigs, then we were dealing with terabytes, and now soon we're dealing with petabytes. And so what we're really looking at NVIDIA is how to handle this volume of data and do it in a very fast and cost-effective way. And with that, we have this sort of virtuous cycle of what is genomic analysis or next-gen sequencing analysis. And it is very much a circle where you start with the sequencing, you generate your data, you align to a genome, do your variant callings, annotate, rinse and repeat, and do it over and over again. With that, one offshoot of that is always to improve the reference, and then along the way improve all the steps within this. So we're looking at a very computationally intensive workflow with big data, and then on top of that, a lot of noise in the system. There was actually a lot of noise in the alignment, mapping to the reference. Sometimes the reference is not complete for the study at hand. Genetic variants, and a lot of time spent in today's presentation talking about the noise in calling genetic variants, and then annotations. So at NVIDIA, we're trying to simplify this workflow, make it faster, and industrialize it. And I think today's project definitely shows some of the outputs of that work. On the next slide, just sort of a tangent, but just a reminder that there is a carbon footprint for what we're doing here. This is on the left-hand side, a bioRxiv publication that came out last year. And you can see that there are substantial carbon footprints for just running a simple data analysis. So you can see it's 4 to 5 years tree life for one RNA analysis. So that's a tremendous amount of carbon footprint to do bioinformatics. And then on the right-hand side, just sort of very similar to what Will was talking about, not only are we faster in providing the accuracy, but we're much more economical, not just in the pricing, but in the power utilization. And you can see that using a GPU actually helps to reduce your carbon footprint. Now on the next slide, we have the full stack of what Parabricks looks like. We support germline, cancer studies, and also RNA. And on the next slide, you can see that just a sampling, not all the tools, but most of the tools to support these pipelines. So whether you're using molecular barcodes for gene panels on the left side, or just going straight forward with a simple FASTQ, have all the tools that allow you to pipeline everything to get to that VCF, and then a whole suite of different variant callers to extract as much information as possible from that data. Now, on the next two slides, we'll talk about germline because that's most relevant for the project at hand. Using the workflow shown on the left-hand side, you can see our compute times in the middle, where effectively we can do a whole human genome at 30x coverage in less than 30 minutes. And we just seem to get faster and faster at NVIDIA with that, reducing the time to analyze these large data sets. And on the right-hand side, you can see that we do support haplotype caller and deep variant, and the F1 scores are multiple nines. So we're able to be faithful to the algorithms, the original algorithms, and just accelerating them on that GPU framework. And then the final slide, as we think about doing our variant calling, like this idea of applying multiple variant calls and extracting all the information possible. What we're looking at here is HG002 and a comparison between deep variant versus haplotype caller. So on the left-hand side, we have a upset plot, and you can see for the vast majority, the variants called are shared between haplotype caller and deep variant. But in the second column over, you can see that haplotype caller has quite a few false positives, or what appear to be false positives relative to the truth set in the top. And the third column over, you can see that deep variant is calling more true variants than haplotype caller has missed. So on the right-hand side, you can see just a sampling of how you may be able to apply both variant callers, where you could either do the union or the intersection. So the union, you want to catch everything, and then that you're going to suffer through a high false positive rate, but a low false negative rate. Or counter, if you need your data to be more stringent, more conservative, you can look at the intersection, where your false positive rate is substantially reduced, but the counter then will be a higher false negative rate. So just a sampling of what Parabricks is and the tools we have, and how then Regeneron has applied this to the UK Biobank. So with that, I'll turn it over to Ben, who's going to show you how to access all of this.
Thanks so much, Tim. I'm obviously biased on this one, but I'm always blown away by what GPUs are capable of. It's great to see them being applied to rapid analysis like this. Yes, with that, I'll hand it over to Ben Busby of DNAnexus, who's going to give us a walkthrough on how to rerun the analytical pipelines. Over to you, Ben.
So I have 7 or 8 minutes to go through how to run this pipeline. So I'm going to try to go through as quickly but as thoroughly as I can. If you have additional questions, please keep putting them in the chat. And if you have questions that occur to you after the webinar, or if you're watching this on a video, please check out community.dnanexus.com. It's a place where I hang out, answer questions, that kind of thing. So feel free to post questions there. I work for DNAnexus. And I'm excited to talk about how to run the pipeline that the Exome consortium has come up with. And I really want to make sure everybody understands that you can run this on the UKB research analysis platform, or if you're a DNAnexus customer, you can run the same pipeline on DNAnexus per se, on the DNAnexus platform. So, for example, if you wanted to bring your own data, as Will was talking about, but it wasn't directly related to your UKB application, that's fine. You can bring it onto the platform at DNAnexus, run this same pipeline on it. So that's just a little bit about DNAnexus. And for those of you who aren't familiar with DNAnexus, we really have a bunch of packaged apps and workflows on DNAnexus, and you can also build your own, bring your own, that kind of thing. Build your own with our wizard, bring them in through Whittle. There's a whole bunch of options there. And so if you look through the apps we have, all of the apps I'm about to talk about are right there, and they're prepared for you. And as Will alluded to, on the research analysis platform, as well as the DNAnexus platform, you can tweak the compute parameters for whatever works for you. So that's great. Now, really starting off, Will talked a lot about the OQFE protocol, and I suggest just Googling for Exome Consortium OQFE protocol. The paper's right there. When these slides are updated and released, there'll be a link right on this slide. And so you're going to use that for mapping. Now, if you're using the UKB research analysis platform, you will find that the CRAMs you get are already mapped to GRCH38 with alts. And if anybody ever wants to talk about why alts are super important, great. Post something on community.DNAnexus.com or on Twitter, and I'll be happy to alert the GRC. But anyway, so yeah, so you'll find these CRAMs already mapped, and that's why I'm gonna jump over the OQFE protocol, but that will went into detail. And then what you're gonna do next, what you want to do here to be consistent with the Exome Consortium is called the Parabricks Accelerated Deep Variant Pipeline. Now, here I'm showing you how to run this through a GUI, but I'd like to point out that on the research analysis platform, as well as the DNAnexus platform, you can run basically everything you can do in the GUI, you can do on the command line and vice versa. That makes it really nice if you're collaborating with a number of folks, biologists, clinicians, all of that. So anyway, so what you're going to do is you're going to go ahead and run this through Deep Variant, as Will and Tim mentioned. And I just like to point out here that if you're just getting your feet wet, you're starting to mess around with this. You don't have to take tons and tons of mapped exomes and start messing with them. You can just take a small sample, and it's not super expensive, and you can always back out. So it's easy to terminate your runs on the GUI, on the command line. But also, if you do a small run, I think I grabbed 3, 4, 5 CRAMs, something like that. We're talking about something like 10 pence in 4 minutes. So really, I would encourage all of you to come in here and really explore with Deep Variant, particularly on the UKB research analysis platform. There are really, really beneficial rates negotiated with AWS and UK Biobank. So really, I think you should feel like you can check those out. You have some freedom here to explore in your research sense and do a bunch of the things that Will was talking about. So then, after I map with Deep Variant, then what I want to do is think about the regions that I want to consolidate from the GVCFs into the PVCF. And here what I'm doing is I'm using bed tools, and I'm taking flanks of the target region. So I want the target regions for the exome capture plus 100 nucleotides on each side. Quite easy to do with bed tools flank. Most of you are probably familiar with that. If you're not, Google with it. But here at DNAnexus, I'm using that inside our Swiss Army knife tool. And that's really a multi-purpose tool that's got a whole bunch of apps we use every day. SAM tools, Plato, all kinds of stuff that VCF tools, BCF tools, that you might use all the time for bioinformatics analysis. And those can be used within this Swiss Army knife tool, again, either on the GUI or on the command line. So that's really convenient. Also, if you wanted to build onto this and add some more of these common bioinformatics tools, you could do that. So that's all an option. Just reach out if you're really not sure sort of how to get started, and we'll help you out with that. And then when I run these apps in the GUI, I just put the command line right there in where it says command line. On the command line, it's -ICMD, and I'll put the same thing on the command line. So that's great. And then I use this really super scalable VCF merger called DNAnexus. And this is open source. You can use it anywhere. As Will mentioned, we really want to make all of these things fair. So if you want to use DNAnexus somewhere else, you can do that, grab it from GitHub, but it is sort of configured and ready to use on both the UKB research analysis platform as well as the DNAnexus platform. So that's really quite nice. And you can grab that. And I really want to mention that this is scalable and it's easy to scale. And that's a big deal for a lot of the folks I talk to. I mean, of course, you can use SAM-Tools merge for, pretty small data sets. But when you're scaling up to bigger things, DNAnexus, in my opinion, is a really nice tool. And you can check it out on GitHub. And then, so what I want to do when I use DNAnexus is I want to make a manifest. So I can do that jumping on the command line, and there'll be step-by-step instructions for this. I'll show you a link in a minute. By jumping on the command line, using a dxfind command bounded by a project, you'll see how to do that. Well, there's going to be some examples in the tutorial. But you want to grab a number of these GBCFs, and then you're going to merge them into PVCF in the regions that we talked about from BenTools. So yeah, you can run DNAnexus, and you can run DNAnexus on actually pretty small groups. Now, Will talked about what it looks like to merge, say, 200,000 exomes, but I would imagine most of you aren't remerging 200,000 exomes. You probably are interested in a specific disease, a specific subcohort, something like that. If you are interested in merging data at that scale, please reach out. Please feel free to reach out if you want to do that, either on the DNAnexus platform or on the research analysis platform. And we'd really love to make your data more available to other people. In my personal view, I think it's really important that disease researchers are able to get as much data as they possibly can. So once you do that, then the ball is really rolling downhill. And we can use BCF tools to take that merge data and process it, as well as convert it to plate format. So that's really quite easy to do. And again, all of this will be in the step-by-step instructions. So just relatively easy to run. And the point of sort of my talk here is really to encourage you to go in and explore both these data sets and other data sets that you might want to bring in and merge with them. So step-by-step documentation, as I mentioned, is available at this link. You can check it out. Check out the Git book for the UKB research analysis platform. I find it super useful. It's also pretty concise. So if you're a computational biologist, bioinformatician, you don't really like to read sort of super verbose stuff, and you want to really jump into things, it's a great resource. I mentioned community.DNAnexus.com a few times. Try jumping on that. Anybody can jump on there, get an account, talk to other bioinformaticians. If you're a novice person using the wrap, it's a great space. Sort of think of kind of a Stack Overflow or a BioStars specifically geared towards the UK Biobank research analysis platform. Or if you're interested in a demo of DNAnexus Apollo and all of these functions plus imaging and so on, go ahead and check that out. With that, I want to leave just a couple of minutes for the Q&A. So I'm going to kick things. First, sorry, I can't skip this. I'd really like to thank Yiqi Huang and Ames Ma, who put together those step-by-step instructions that I'm hoping you use, as well as the computational biologists and Will's team at the RGC, some of them old friends of mine, for putting the consensus pipeline together. With that, I'm going to kick it over to Harry. I know we have some really good questions. I've been answering them, so I look forward to this sort of short panel discussion.
Fantastic. Thank you so much, Ben. That was really great, really helpful stuff. And thanks to all the speakers. I think they've been fantastic talks all around. So yeah, let's take some questions. I'm going to start with one for Mark. Mark, what is the timeline to release the Nightingale metabolome data that you had?
That's a good question. So as I mentioned, we've made available the first 120,000 measures in March last year. We don't have a formal timeline for the remaining data at this point. The assays are underway as we speak. I'd anticipate that there'll be a follow-up release sometime in 2023, so sometime next year, and then I'm sure the remaining data sometime thereafter.
Thank you very much. I have one for Will. Will, are there going to be more Exome releases on the UK Biobank platform on RAT?
Yeah, there will be, as Mark mentioned previously, there will be the balance of the samples. We're at 450. We're going to get as close to that 500 as we can, and that release we're targeting this year, which will be processed the same way, and it'll be an aggregate release. It won't just be the delta. And then, people should keep their eyes out, because as the community does process these data at scale, UK Biobank will do a great job of returning some of those other data sets out there. So there will be more samples coming. We're targeting this year as an aggregate release. And then if people are asking about other types of data that are on those exomes, it's a continual process. So looking forward to that.
Fantastic. One question for Tim here. Tim, are you planning on extending Parabricks functionality on DNAnexus?
Depending upon customer requests, we can definitely extend the functionality, working with all our partners here on the call.
Fantastic. I have one for Ben, if I have an approved UK Biobank project, how do I get access to the interface you showed?
Oh, right. So yeah, just go to ukbiobank.dnanexus.com and sign up. It should be a really quick process, and then you should have access. When you get into the interface, and there's instructions that I've very recently put on community.dnanexus.com, but make sure you click the box to dispense the data when you set up a new project, and that will get all your data up there and ready to go, as we've been outlining in this webinar.
Nice, thank you. This is great, lots of questions coming in. Another one for Mark. Mark, how are relatives handled? Are there pedigrees in UK Biobank data?
So yes, I guess as part of the original genotype release, there's a lot of PCA analysis. We don't have field level data which determine the relatives, but there are a number of trio samples in there. And I mentioned about the serology study. We have actually gone as part of that to request support from relatives, children and grandchildren. Those assay data are available, but they're effectively being treated separately.
Got it. Thank you. One for Will here. It's a request for another webinar. Are you going to host a webinar for downstream analysis of the variant calls? So functional annotation, querying the variants, and correlating variants with other phenotypes.
I think we'd love to set that up. It wouldn't be me. We would get the experts on those steps in. But I know that there is a great deal of documentation and work. Sheldon Bai on the RGC team, who did a lot of the documentation work on this protocol, has worked with our downstream teams on genotype, phenotype, and Regini, and a lot of those types of association work. Those protocols are all going to be available on the UKB RAP documentation site. And I think having a webinar on that would be great. So we will take that feedback and see what we can do.
Fantastic. One for Ben, which is a bit of a technical question. Can I look at homozygous reference genotypes in WES regions using a bed file? Or do you have to convert to GVCF? And then are the homozygous genotypes included in the output PVCF?
That is a great question. And I will say that I've looked at it a couple of times and I'm not entirely sure. So tell you what, if you put that question on community.dnanexus.com, I will figure that out, i.e. ping Mike Lynn, or try it myself and get back to you in the next couple of days.
Great, thank you. One more for Mark. Mark, it's great that UK Biobank's working with DNAnexus to put in place a platform that allows researchers to analyze these large-scale data sets. Is there any provision for researchers' costs to be covered, given analyses of these data can consume considerable compute and storage?
Okay, that's a good question. I'm sure maybe a concern to some of the people on the webinar today. Perhaps a couple of comments. So the first is that all of the biobank data, of which there's about 15 petabytes so far, is held as a sponsored data set within the platform, which means that researchers aren't paying for that. They only pay for the storage of data that they derive. The second, I guess, is DNAnexus are kindly providing £40 worth of research credits to allow people to get familiar with the platform, so they can get used to how the platform works before they start incurring costs for their research. And again, I think it's already been mentioned, the competitive rates provided by Amazon. I think the tools we've talked about today provided by NVIDIA really help to undertake cost-effective analyses. And perhaps the third is just to reference a program that we've gone live with just in the last month, which again is courtesy of Amazon. We're able to provide $500,000 of research credits each year to early career researchers and those researchers in low and low middle income countries. So please do go and look at the Biobank website for more details on that, and really keen to use those credits and to give them to eligible researchers as best we can.
Fantastic. And then one for Will, are there short tandem repeat calls available in the data set?
Not in the set that we release now, in the Exome data. And I think if there's interest, this feedback is great. I would encourage people though to keep an eye out on the whole genome data as that becomes available, because I think there's going to be a lot of robust, large, complex variation that can be very well captured in those data they're released. So as you see those, please take a look.
Perfect, thank you. I think that's most of the questions. A couple more around sort of getting this step-by-step guide into people's hands. So once again, yeah, the recording and the slides from the session are going to be made available by DNAnexus on the Resource Center. And I think that's everything.
Yeah, thank you. Thank you all for joining today. As Harry said, we'll be circulating all the links that we referenced today in a follow-on email. And thank you for joining. Have a great afternoon, evening, and we'll see you next time.